So here we're going to go over lactic acid fermentation. So this process is going to occur in anaerobic conditions. What does this mean? It's going to occur when we have no oxygen. Now, an important thing to understand is that this process begins with pyruvate. Now, where does this pyruvate come from? Remember that this pyruvate is coming from glycolysis. Now, glycolysis itself is an anaerobic process. It does not require oxygen. So after we get the production of pyruvate from glycolysis, it has three different fates. It can go on to the TCA cycle, and then it will go on to the ETC cycle, and we'll get a lot of ATP generated through oxidative phosphorylation. Or this pyruvate can go on to lactic acid fermentation, which we're going to go over right now. Or it'll go on to ethanol fermentation. And between lactic acid and ethanol fermentation, it just depends on what organism or what cell you're going to be talking about. So lactic acid fermentation, it can actually occur in animal cells, specifically our muscle cells. So whether we go into fermentation or whether we go into the TCA and ETC, it all depends on whether or not we have oxygen. When we don't have oxygen, we're going to do fermentation. When we do have oxygen, we're going to go into the TCA and ETC. Now, it is more favorable to go into TCA and ETC because we get a lot more ATP produced this way. Fermentation does not produce as much ATP. But the whole purpose of fermentation is to regenerate NAD+. Why do we want to regenerate NAD+? Well, we have to remember that in glycolysis, when we're breaking down glucose, we have a step in glycolysis that reduces NAD+ and produces NADH, because this NADH goes on to the electron transport chain. But if we cannot go down to the electron transport chain because we don't have oxygen, we're in anaerobic conditions, we're going to have this reduced NADH from glycolysis, and we have to regenerate it to NAD+, so it can be used again in glycolysis to have this glycolysis keep on moving forward. So essentially, we have a specific pool of NAD+, and we have to regenerate it to at least allow glycolysis to continue to get some ATP production. So lactic acid fermentation itself is actually a simple step. We have this pyruvate from glycolysis, and we're just going to reduce pyruvate to yield lactate. So pyruvate gets reduced to lactate, and NADH will get oxidized to NAD+, and the enzyme that we're using is lactate dehydrogenase. Now let's go over here and think about this one more time. So we have glucose, and this glucose is going to be broken down via glycolysis, and glucose is a 6-carbon molecule, and it gets broken down into two 3-carbon molecules. So it's going to get broken down into 2 3-carbon molecules. What are those 3-carbon molecules? Well, we're talking about pyruvate because it's a 3-carbon molecule, and at the end of glycolysis we get 2 pyruvates. So those pyruvates can go on to produce lactate. So for every 1 glucose, we will get 2 lactates produced. And also remember that glucose produces a net yield of 2 ATP. That is why we have 2 ADP, which will combine with 2 inorganic phosphates, and we'll see the production of 2 ATP. So through glycolysis occurring, we get 2 ATP, and through fermentation, we regenerate that NAD+, which is going to be fed back into glycolysis to regenerate that 2 AT+. So the 2 ATP. So the whole purpose of fermentation, once again, is to regenerate NAD+, so it can be used in glycolysis to produce that ATP. And another last thing we have to make note of is at the end of the day, there is no net oxidation or reduction, because in glycolysis, we reduced NAD+ to NADH, but then in fermentation, we oxidized NADH to NAD+, so we have the same end result. So there was no net oxidation or reduction. And that is essentially lactic acid fermentation.